All right, get ready to dive into a world that's teeming with life, but mostly invisible to us. We're talking prokaryotes, those single-celled masters of the universe, the bacteria and archaea.
And they're not just small, they are everywhere. I mean, trillions and trillions, just these little guys inhabiting every nook and cranny you can think of on the planet.
Our chapter excerpt really drives that home, doesn't it?
Oh, yeah.
Did you know that there are more prokaryotes in a handful of soil than there are stars in the entire Milky Way galaxy?
It's pretty amazing. And what's really wild is their ability to thrive in environments that we couldn't even imagine. Boiling hot springs, those crazy salty lakes, even near those deep sea volcanic vents where temperatures can reach, hundreds of degrees
It's
Celsius.
like they're from another planet. Okay, so let's get down to the basics. What sets prokaryotes apart from our own cells, eukaryotic cells?
Well, for one, they're much simpler. They don't have a true nucleus, so their DNA isn't neatly packaged, within a membrane like ours.
So where is it? Just floating around.
Not exactly. It's concentrated in a region we call the nucleoid, but it's not enclosed in a membrane. And unlike our multiple linear chromosomes, prokaryotes typically have just a single circular chromosome.
Ah, compact and efficient. The chapter did mention a giant exception to this rule, though, a bacterium so big you can see it with the naked eye.
Yeah, Theomargarita namibiensis. It's a sulfur-oxidizing bacteria found in ocean sediment. Definitely an outlier in the prokaryotic world.
So, size can be deceiving in the prokaryotic world. How do these tiny cells manage to conquer such extreme and diverse environments? What are their secrets?
Well, one key factor is their cell wall. It provides structure and protection and prevents the cell from bursting.
Right, and that's where the Gram stain comes in, right? The chapter mentioned that it's a key technique for differentiating bacteria based on their cell wall composition.
Exactly. It's named after Hans Christian Gram. Back in the late 1800s, he developed this technique that we still use today because it's so useful for diagnosing and treating bacterial infections.
So how does it actually work?
It's all about how the cell wall structure reacts to certain dyes. Gram-positive bacteria, with their thick peptidoglycan layer, they hold onto this crystal violet stain, so they appear purple under the microscope.
And gram-negative bacteria.
They've got a thinner peptidoglycan layer and an outer membrane, so they end up staining pink.
So it's not just about pretty colors. This staining technique actually has real-world implications for medicine.
Absolutely. It helps doctors determine which antibiotic will be the most effective.
How so?
Well, the outer membrane of gram-negative bacteria, it acts like a barrier, making them more resistant to certain antibiotics. And that outer membrane also contains these things called lipopolysaccharides, which can be toxic, and they can cause a big immune response in our body. So that can lead to complications like fever and shock.
So a simple staining technique can tell us a lot about a bacterium's defenses. Pretty incredible. The chapter also mentioned that some bacteria have even evolved resistance to antibiotics that we hadn't even discovered yet. How is that possible?
It's pretty interesting. Bacteria have been fighting their own chemical wars for millions of years, producing substances to inhibit or kill other bacteria.
So some bacteria had these defense mechanisms already in place, and then we come along and start using them as medicines.
Exactly. We've kind of stumbled into this ancient battle.
And unfortunately, we've accelerated things by overusing and misusing antibiotics, which has given resistant bacteria a huge advantage.
That's right. It's a serious problem. But for now, let's talk about some of the other ways prokaryotes have adapted to survive.
The chapter highlights some pretty amazing ones. Capsules, slime layers, endospores. It's like they have this whole arsenal of survival gear.
Exactly. And each one is specific to a particular challenge. For example, capsules. They're sticky outer coats that help bacteria stick together, or to surfaces to form those microbial communities we call biofilms.
So they're kind of like these microbial cities all huddled together.
Yeah, and these capsules also help bacteria evade our immune systems, which is why some can become pathogens. Sneaky. Then there are endospores. Talk about hardcore survivalists.
Endospores are like little microbial bunkers. Some bacteria form them when they encounter really harsh conditions. They can withstand things like boiling water, extreme dryness, even radiation.
The chapter mentions Bacillus anthracis, which causes anthrax. Its endospores can survive for decades in the soil. What's even going on in there?
It's like hitting the pause button on life. They make a copy of their chromosome, encase it in a multi-layered structure, get rid of most of their water, and basically shut down metabolism.
They get dormant.
Exactly. And they can stay that way for incredibly long periods. When conditions improve, they rehydrate and start up again.
Like a built-in time machine. That's amazing.
It is. And while some prokaryotes are masters of hunkering down, others are incredibly mobile, they can zip around at surprising speeds.
Yeah, the chapter talks about taxis, how some prokaryotes move toward or away from a stimulus. They actually have a sense of direction.
That's right. For example, they can respond to chemical signals, moving toward nutrients or away from toxins. And they can really move. They mentioned one that can move 50 times its body length per second.
Like a human sprinting at over 200 miles per hour. They manage this with flagella.
Those whip-like structures that help cells move.
Right. Both prokaryotes and eukaryotes have them, but they work really differently. Prokaryotic flagella are thinner and rotate like propellers driven by a tiny molecular motor.
So intricate. Speaking of complex, let's talk about the inner workings of prokaryotes.
All right. Now remember, prokaryotic cells may be simpler than our eukaryotic cells, but they're not just empty sacs. They have their own internal organization and specialized structures to carry out all the functions of life.
Right, they have those specialized membranes.
Exactly. They don't have organelles like mitochondria or chloroplasts, but they can have special membranes for specific functions like respiration or photosynthesis. Often these are folds in the plasma membrane.
What about plasmids? We talked about them earlier.
Yes. They're like little bonus loops of DNA, separate from the main chromosome. They often carry just a few genes, but those genes can be very important. For instance, they can provide resistance to antibiotics or allow the bacteria to break down unusual nutrients.
So plasmids are like genetic add-ons.
Yeah. And the really cool part is that they can be transferred between bacteria through this process we call conjugation.
Conjugation. Is that like bacterial.romance?
It is often referred to as bacterial sex, but it's not quite the same as how eukaryotes do it. It involves directly transferring DNA from one bacterium to another through a structure called a pilus.
They're literally sharing genetic material.
Yes, and that shared DNA can include those plasmids. This ability to share genes is why bacteria can evolve and adapt so quickly.
Wow. It's both cool and kind of scary, like they have their own underground information network.
It is a remarkable example of how microbes cooperate and adapt, and it has huge implications for things like the spread of antibiotic resistance. But before we get into that, let's talk about how prokaryotes reproduce.
They primarily use binary fission, right?
Yep. It's basically asexual reproduction, where one cell divides into two identical daughter cells.
Like cloning themselves.
Pretty much, and it can happen so fast. In ideal conditions, some can divide every 1 to 3 hours, a whole new generation before you've even finished lunch. That's why they can colonize environments so quickly.
That's a population explosion.
Yeah.
But doesn't that limit their genetic diversity if they're all clones?
That's where mutations come in, those random changes in the DNA sequence.
And even though mutations are rare, they can add up quickly with short generation times and huge populations.
Exactly. It's all about the numbers. A small mutation rate can still generate significant diversity when you have trillions of individuals reproducing multiple times a day.
They used E. coli as an example. Estimated that there could be over 8 million new mutations every single day in the E. coli in a human intestine alone.
That's a lot of potential for evolution. It shows how quickly they can adapt. The chapter even mentioned E. coli evolving to thrive in a low-glucose environment in just 20,000 generations.
So even small changes can add up to big adaptations over time.
It's a powerful demonstration of how evolution works.
Okay, so we've got rapid reproduction, mutations happening all the time, and even genetic swapping. Sounds like they have a lot of ways to mix things up.
They do, and that diversity is a big part of why they're so successful. It lets them adapt to such a huge range of environments, from the bottom of the ocean to inside our own bodies.
Speaking of diverse environments, they don't all rely on the same food sources, right?
Not at all. Prokaryotes have an amazing range of ways to get energy and carbon. They even outdo eukaryotes like us.
The chapter breaks it down into 4 major modes of nutrition: photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. That's a lot to remember.
It seems complicated, but it really comes down to 2 simple questions. Where do they get their energy from, and where do they get their carbon from?
So photoautotrophs are like plants. They use light for energy and carbon dioxide for carbon.
Exactly. Then there are chemoautotrophs. They use inorganic chemicals for energy, things like hydrogen sulfide or ammonia, but they still use CO2 for carbon.
Like super recyclers, turning inorganic compounds into organic matter. Pretty impressive. What about photoheterotrophs?
Now, those are interesting. They use light for energy, but they get their carbon from organic compounds. You only find this in certain prokaryotes, especially those in aquatic environments.
A hybrid approach.
Right.
And finally, chemoheterotrophs.
They're the ones most like us, using organic compounds for both energy and carbon. It's a pretty common approach.
So they've got all the bases covered.
They do, and their metabolic diversity doesn't stop there. There's also a range of adaptations when it comes to oxygen. Some need oxygen.
Like us.
Right, those are obligate aerobes. But others, called obligate anaerobes, can't tolerate oxygen at all. It's actually toxic to them.
Really? How can anything survive without oxygen?
They've evolved other ways to get energy, like fermentation and anaerobic respiration. They use other molecules as electron acceptors.
Wow. So they can thrive with or without oxygen. Amazing.
Right. And then there are facultative anaerobes, which can switch between using oxygen and using those other pathways depending on what's available.
Like they have a backup plan for everything.
Exactly. Prokaryotes are masters of flexibility and resourcefulness. That's a big reason why they're so successful.
My mind is blown. We've covered so much. Structure, adaptations, metabolism. What else is there?
We're just scratching the surface. There's a remarkable array of lineages, each with its own unique characteristics.
Now I'm intrigued. Tell me more about these different prokaryotic lineages and how we even make sense of such a diverse world.
Well, before we get into specific lineages, we need to talk about the revolution that's been happening in how we classify these organisms. It's all about molecular systematics.
Molecular systematics. That's
Right.
using DNA to figure out relationships, right?
Yes. It's not just about physical characteristics anymore. Those can be misleading with microbes. Now we analyze DNA and RNA sequences to figure out evolutionary relationships.
And how has that changed our understanding of prokaryotes?
Well, back in the 1970s, scientists started using ribosomal RNA as a marker for these relationships. And guess what? A lot of prokaryotes that we thought were bacteria were actually more closely related to eukaryotes. This led to a whole new domain, archaea.
Wow. A whole new domain of life.
It is. And our understanding of prokaryotic diversity keeps expanding thanks to techniques like PCR and metagenomics, where we can study the genetic material of whole microbial communities from the environment.
Right. PCR is like a molecular copying machine for DNA. And metagenomics takes it a step further. We can analyze DNA from those communities without even growing them in a lab.
You got it. These techniques have been revolutionary. It turns out there are way more prokaryotic species than we ever imagined.
The chapter mentioned that while we've only named about 16,000 prokaryotic species, a single handful of soil could have 10,000 different species.
It's mind-boggling. We're just starting to understand the true extent of their diversity. And it's not just the number of species, it's also the incredible range of adaptations.
So how do scientists even categorize these lineages? What are some key features that set them apart?
Well, within the domain bacteria, we have tons of groups, each with its own characteristics. There are the proteobacteria, which are incredibly diverse. They include photosynthetic species and some that cause diseases like cholera.
And the cyanobacteria, those oxygen-producing bacteria.
Yes, they're fascinating. They're thought to be the ancestors of chloroplasts.
So they're the original oxygen producers.
Exactly. They had a profound impact on the planet's atmosphere and how life evolved.
Incredible. What other major bacterial groups are there?
Well, there are the gram-positive bacteria, which includes some you've probably heard of, like Staphylococcus aureus.
That's the one that causes staph infections, right?
Right. And the ones that stain purple with the Gram stain. And then there are the spirochetes. They're spiral-shaped and they move in a corkscrew motion. Them are harmless, but others cause diseases like syphilis and Lyme disease.
So bacteria can be everything from beneficial oxygen producers to harmful pathogens. What about archaea? What makes them so unique?
Archaea are really interesting. They share some traits with bacteria, but they also have characteristics that make them really stand out. And they're often found in the most extreme environments on Earth.
Like the extremophiles lovers of extreme conditions.
Exactly. They can survive in places that would be lethal to most other life forms. We have the extreme halophiles, which live in extremely salty environments, like the Dead Sea or that pink lake in Spain.
And the extreme thermophiles. They thrive in boiling hot springs or near volcanic vents.
They do. They've evolved ways to protect their proteins and DNA from those scorching temperatures.
They mention one that can even reproduce at 121 degrees Celsius, hotter than boiling water.
Yeah, strain 121. It lives near deep sea vents. It's amazing how resilient life can be.
It is,
Yeah.
but we often think of archaea as extremophiles, but a lot of them actually live in more moderate environments too, right?
Yes, they're in soils, lakes, even our own guts.
There are archaea in our guts.
Absolutely. Methanogens, for example, are archaea that produce methane, and some live quite happily in our intestines, part of our gut microbiome.
I had no idea.
We are walking ecosystems.
Okay.
But let's get back to the lineages within archaea. We talked about Euryarchaeota and Crenarchaeota as two main clades.
And you mentioned that new discoveries have changed our thinking about archaeophylogeny.
Yeah, remember the tax supergroup. And those Lokiarchaeotes, which might be the closest relatives to eukaryotes.
Yes, the potential missing link between prokaryotic and eukaryotic cells.
Precisely. They have this really cool mix of characteristics, some bacterial, some eukaryotic, and some totally unique, like a genetic mosaic.
So studying them could help us understand how eukaryotic cells even evolved.
Exactly. They could hold the key to one of the biggest mysteries in biology. How those simple prokaryotic cells gave rise to the complex eukaryotic cells we see in all plants, animals, and fungi.
The archaeal world is full of surprises, but let's zoom out for a second. Why should we care about prokaryotes at all? What's their role in the big picture?
Prokaryotes are absolutely essential to life. They're the unsung heroes of the biosphere, they're critical for nutrient cycling, and even the very existence of entire ecosystems.
Okay, tell me more about how something so tiny can have such a big impact.
Well, think about chemical recycling. The atoms that make up all living things, including us, come from the soil, air, and water. Eventually, those atoms will return to the non-living environment.
And how do prokaryotes fit in?
They're the ones breaking down dead organisms and waste products, releasing those elements back into the environment. They're nature's recyclers, keeping the cycle of life going.
So without them, things would just grind to a halt.
Exactly. They keep the planet's ecosystems functioning and make sure there are enough nutrients for all life.
Impressive.
And their role in recycling isn't just about breaking things down. They also convert certain molecules into forms that other organisms can use.
Oh, like what?
Well, cyanobacteria make sugars through photosynthesis. Those sugars then get passed up the food chain.
The basis for lots of food webs.
Exactly. And many prokaryotes can fix nitrogen. They convert atmospheric nitrogen into forms that plants can use for proteins and nucleic acids.
So not just recyclers, but producers and transformers of essential nutrients.
Exactly. Like the master chefs of the biosphere, making sure everyone gets what they need.
Okay, that's a good analogy. What about symbiosis? You mentioned earlier that prokaryotes are masters of symbiosis.
They are. They form all sorts of relationships with other organisms, from mutually beneficial partnerships to those that can be harmful.
The textbook gave the example of our own gut microbiome, all those bacterial species helping us digest food and even influencing our immune system.
That's a perfect example of mutualism, where both partners benefit. They get a place to live and food, and we get help with digestion and protection from harmful bacteria.
So we really depend on those little guys for our health.
We do, and prokaryotes have tons of symbiotic relationships with other organisms too, all over the world.
The chapter even mentioned that some ecosystems completely rely on prokaryotes, like those hydrothermal vent communities.
Those are such great examples of how life can exist in the most unexpected places, fueled by chemoautotrophic bacteria.
Chemoautotrophic, those are the ones that get energy from inorganic chemicals, right?
Exactly. They don't need sunlight. In this case, they use chemicals released from the vent, like hydrogen sulfide.
So they're the foundation of that whole ecosystem.
They are. Without them, those incredible deep-sea communities wouldn't exist.
Amazing. It really shows how interconnected everything is.
It's true. Everything is part of a complex web, and even the smallest organisms are important.
Okay, so we've talked about the good side of prokaryotes, but we can't forget about the bad guys, those disease-causing bacteria.
You're right. Most prokaryotes are harmless or even beneficial, but there are some that can cause harm.
And those are all bacteria, right? No archaeal pathogens.
As far as we know, yes. All known pathogenic prokaryotes are bacteria, and they can cause all kinds of diseases, some of which are really deadly.
Like tuberculosis, which still kills millions of people every year.
It does. And diarrheal diseases, which are especially bad in areas without clean water and sanitation.
The chapter talked about how bacteria cause disease. They often do it by producing toxins.
Right. There are two main types: exotoxins and endotoxins.
Exotoxins are the ones that are secreted by the bacteria, right?
Yes, and they're often proteins that disrupt cell function or damage tissues.
Cholera and botulism are both caused by exotoxins.
That's right. The cholera toxin causes severe diarrhea and dehydration, and botulism toxin can paralyze muscles, and it can be fatal.
Sounds pretty scary. What about endotoxins?
They're different. They're part of the outer membrane of gram-negative bacteria.
So they get released when the bacteria die.
Exactly. They can trigger a strong immune response, which can cause fever, inflammation, and even shock.
Like with salmonella, right? That
Yeah.
can cause food poisoning.
Yes, salmonella can cause a lot of unpleasant symptoms.
Okay. So pathogenic bacteria cause all sorts of trouble with their toxins. But I think the most worrying thing is antibiotic resistance.
It is. It's a huge public health threat, and it's happening because we've overused and misused antibiotics.
And bacteria can evolve resistance so quickly. The chapter said sometimes within a few years of a new antibiotic being introduced.
It really is natural selection at work. When we use antibiotics, we're basically favoring the bacteria that already have resistance genes.
So they survive and reproduce, passing those genes along.
Right. And to make things worse, they can share those genes through horizontal gene transfer, like conjugation, which we talked about earlier.
Like they're sharing a secret weapon.
Yeah, that's why we're seeing more and more multi-drug resistant bacteria, which are really hard to treat.
They use the example of XDR-TB, extensively drug-resistant tuberculosis, which is a really serious problem.
It shows just how bad antibiotic resistance can get. And it's not just TB. We see it in a lot of bacterial pathogens, making it tougher to treat infections.
So what can we do? Are we doomed to a future without antibiotics?
It's a challenge, but we're not doomed yet. We have to be more careful about when we use antibiotics, both in medicine and agriculture.
So only use them when absolutely necessary and take the full course, even if we feel better.
Right. And we need more research to develop new antibiotics and new ways to treat bacterial infections. There are some promising new developments. The chapter mentioned melacidins, which are a new class of antibiotics that have been effective against multi-drug resistant pathogens.
That's good news. Are there any other promising leads?
Yes, some scientists are looking at using bacteriophages, which are viruses that infect bacteria, and they're looking into ways to disrupt bacterial communication, which could make them easier to treat.
It sounds like a lot of approaches are needed to tackle antibiotic resistance.
It's going to take a global effort. But for now, let's end on a positive note. Prokaryotes aren't all bad. In fact, they can be very useful. The chapter talked about some of the ways they're being harnessed to help us.
Yes. Beyond their roles in ecosystems, they have a lot of potential for biotechnology and bioremediation.
Absolutely. They've been important in biotechnology and research for a long time, and the more we learn about them, the more potential applications we find.
Like using E. coli for gene cloning and PCR.
Exactly. And now we can genetically engineer bacteria to produce all sorts of things. Vitamins, antibiotics, hormones, enzymes.
And biodegradable plastics. Maybe a solution to our plastic problem.
Right. Scientists are also looking at using bacteria to produce biofuels, like ethanol. There's a lot of potential there.
Sounds like prokaryotes could help us build a more sustainable future.
They could. And one of the most exciting applications is a system that bacteria use to defend themselves from viruses. It's called the CRISPR-Cas system.
CRISPR, I've heard of that. It's a gene editing tool, right?
Yes. CRISPR-Cas9 is a revolutionary technology that could transform medicine, agriculture, so many fields.
And it all comes from bacteria.
It does. It's pretty incredible.
Wow, this deep dive is blowing my mind. We've covered their structure, their amazing adaptations, their diverse metabolic pathways. It's incredible to think about this whole hidden world.
I know. It's like we've been living alongside these microscopic wonders without even realizing it.
And we're just beginning to understand their power and diversity.
Absolutely. And we're still learning so much about how they can benefit us and the planet.
Speaking of that, the chapter also talked about bioremediation. Can you tell me more about that?
Sure. It's a really interesting field, and it's becoming more and more important as we deal with pollution and environmental damage. Bioremediation uses organisms, mostly prokaryotes, to clean up contaminated environments.
So it's like using microbes to fight pollution. What kinds of pollutants can they tackle?
Lots of them. Heavy metals, pesticides, oil spills, even industrial waste. The chapter mentioned using bacteria to break down sewage, converting it into fertilizer.
So they're not just cleaning up, they're turning waste into resources. That's amazing. What about oil spills? Can bacteria really help with those?
Absolutely. Some bacteria love hydrocarbons. They can break those down into simpler, less harmful substances.
It's amazing to think that microbes can help clean up some of our biggest environmental disasters.
It shows the power of nature and how adaptable life is. They've evolved to thrive in so many different environments.
Okay, I want to go back to antibiotic resistance for a minute. It's kind of freaky to think that some bacteria are resistant to things we haven't even invented yet. How does that even happen?
Well, bacteria have been fighting each other for millions of years. They use chemical warfare to compete for resources.
Hmm.
Over time, some have evolved defenses against those natural antibiotics.
So we're basically joining an ancient battle with our synthetic antibiotics.
Exactly. A lot of our antibiotics come from those natural compounds produced by bacteria.
Hmm.
So when we introduce a new antibiotic, we're selecting for the bacteria that already have resistance.
But we're constantly playing catch-up.
We are, and it highlights how important it is to use antibiotics responsibly and develop new strategies to combat infections.
It's a good reminder that we need to keep innovating.
We do. We talked about those archaeal lineages before, the Archaeota and Crane Archaeota, and those recent discoveries that changed our understanding of archaeal phylogeny.
Right, the tax supergroup and those Archaeotes that might be related to eukaryotes.
Exactly.
They're like a missing link.
They could be. They have this fascinating mix of characteristics. It's like they're a genetic mosaic, a snapshot of their unique evolution.
So what can we learn from them?
Studying their genomes could tell us a lot about how complex cells came about. They might help us understand how eukaryotic cells evolved.
It's amazing to think they might hold the secrets to one of the biggest mysteries in biology.
And it highlights how powerful these new molecular techniques are, like metagenomics. We can uncover so much about these hidden worlds.
This has been eye-opening. I have a whole new perspective on the microbial world.
Me too.
It's clear that we share this planet with an incredible array of organisms, and understanding them is crucial for our well-being and for the health of the planet.
That's a great way to put it.
Okay, before we wrap up this section, let's talk a bit more about those biofilms. You mentioned that they can be both good and bad.
Yes. Biofilms are communities of microorganisms that attach to surfaces and create a protective matrix. They can be beneficial, like in wastewater treatment or in the human gut, but they can also be problematic.
Right, they can form on all kinds of surfaces. Pipes, boat hulls, medical devices, even our teeth.
That's right. They can clog things up, corrode materials, contaminate equipment, and even cause infections that are hard to treat.
And they're notoriously resistant to antibiotics.
Yes, the biofilm structure protects the bacteria, making them much harder to kill.
So it's like a microscopic fortress.
Exactly. That's why scientists are trying to find new ways to prevent biofilms from forming and treat the infections they cause.
We've come a long way in understanding microbes, but there's still so much to learn.
It's fascinating, isn't it? They can be both our allies and our enemies.
Absolutely. And speaking of shaping the world, let's talk about some of the practical ways prokaryotes are impacting our lives and the future of the planet.
They really do. It's like we've been living next to this secret world of microscopic wonders, completely unaware of how powerful and diverse they are.
It's true. We've just scratched the surface.
Exactly, and we're still figuring out all the ways they can be used to benefit us and the planet.
Like that concept of bioremediation, right? You mentioned it earlier. Can you expand on that?
Sure, it's a growing field, especially now as we face more and more pollution and environmental damage. Bioremediation is basically using living organisms, usually prokaryotes, to clean up contaminated environments.
So we're enlisting microbes to fight pollution. That's a pretty cool idea. What can they clean up?
You'd be surprised. They can break down all kinds of stuff. Heavy metals, pesticides, oil spills, you name it. The chapter mentioned using bacteria to clean up sewage, turning it into fertilizer.
So they're not just cleaning up, they're turning waste into valuable resources.
Yeah.
That's incredible. Can they even handle things like oil spills?
Absolutely. Some bacteria love to eat hydrocarbons. Those are the molecules in oil. They can break them down into simpler, less harmful substances.
It's amazing that these tiny organisms can help clean up those huge environmental disasters. It gives me hope for the future.
It does. It's a real testament to the power of nature and the incredible diversity of life on Earth. Prokaryotes have been evolving for billions of years, and we can use those adaptations for our own benefit.
Okay, I want to go back to antibiotic resistance for a second. It's kind of unsettling to think that there are bacteria out there resistant to things we haven't even invented yet. How is that possible?
Well, remember, bacteria have been around for a very long time, and they've been fighting each other for survival just as long. They have this whole chemical arsenal they use to compete. And over time, some bacteria have developed ways to protect themselves against those natural antibiotics.
So when we create synthetic antibiotics, we're essentially entering an ancient battle.
That's a good way to put it. A lot of our antibiotics are actually based on those natural compounds that bacteria make. So when we use them, we're selecting for the bacteria that already have resistance mechanisms.
So it's like we're accidentally creating superbugs.
In a way, yes. It's why we have to be so careful about how we use antibiotics. And it's why research into new antibiotics and new ways to treat bacterial infections is so important.
It seems like we're always going to be trying to stay one step ahead.
It's a constant race. But speaking of staying ahead, let's go back to those lineages for a minute. Remember the Archaeota and Archaeota and the Tex supergroup?
Oh, right. And those Archaeotes that might be our closest relatives in the archaeal world.
Exactly.
It's like this missing link in evolutionary history.
They could be, and they're fascinating to study. They have this mix of bacterial, eukaryotic, and unique characteristics. Like they're a blend of different branches of life.
So what can we learn from them?
Their genomes could tell us a lot about how complex cells evolved. It's possible they hold the key to understanding how eukaryotic cells arose from those simpler prokaryotic ancestors.
It's mind-blowing that something so small could contain such important secrets.
It is, and it's thanks to tools like metagenomics that we can even study them. It's opened up a whole new world of discovery.
This deep dive has been a real revelation. I feel like I have a whole new understanding of the microbial world and the impact prokaryotes have.
I know what you mean. It makes you realize that we share this planet with all these incredible organisms and that we need to understand them to protect ourselves and the planet.
Well said. Before we finish up, I want to talk a little more about biofilms. We talked about how they can be both helpful and harmful.
Right, they're those communities of microorganisms that attach to surfaces and create a protective layer. In some cases, they're beneficial, like in wastewater treatment, but they can also cause a lot of problems.
Like the plaque on our teeth.
Exactly, that's a biofilm. And if we don't brush and floss, it can lead to cavities and gum disease.
Not a good thought.
Mhm.
And they can also contaminate medical equipment, which can cause serious infections.
They can, and they're hard to treat because that protective layer makes them really resistant to antibiotics.
It sounds like we need to find ways to prevent biofilms from forming in the first place, or to break them down so antibiotics can work.
That's right. It's a complex problem, but scientists are working on it.
This deep dive has really changed how I see the world. I won't look at soil or even brush my teeth the same way again. It's amazing to think about this whole hidden world of prokaryotes that's all around us, all the time.
It is, and it just goes to show that we still have so much to learn about microbes.
There's so much more than just germs.
They really are. They're essential for life, they offer incredible potential for solving problems, and they're constantly shaping the world around us.
Well said. And on that note, I want to thank you for joining me on this exploration of the prokaryotic world. It's been amazing. And to our listeners, thank you for diving in with us. Until next time, keep exploring, keep learning, and never underestimate the power of the microbial world. So we've covered so much about prokaryotes, their diversity, their adaptations, how they're essential for ecosystems, and how they can even help us solve some pretty huge challenges. But what are some of the real-world impacts? How do they affect our lives in ways we might not even think about?
Well, for one, they're really important for agriculture. They have a big impact on the nutrients in soil, and that's essential for plant growth.
So beyond those nitrogen-fixing bacteria we talked about, there are other prokaryotes that affect how fertile soil is.
Oh, absolutely. Some help make nutrients like nitrogen, phosphorus, and potassium more available to plants, like tiny little farmers.
That's amazing. So they're not just supporting natural ecosystems, but they're also helping us feed everyone.
Exactly. But it's a delicate balance because some prokaryotes can actually make those nutrients less available.
Oh, how does that happen?
They essentially lock up the nutrients, taking them for themselves so plants can't get to them. It's important to understand these microbial interactions if we want to improve agricultural practices and make sure we can produce enough food sustainably.
It's like there's a whole invisible world under our feet, this complex network of plants, microbes, and the soil.
It is, and it's super important for the health and fertility of soil. But agriculture isn't the only area where prokaryotes matter. They impact our lives in so many ways, both good and bad.
Right, you were talking about biofilms earlier. They can be beneficial, but they can also cause a lot of problems.
Exactly. Think about your teeth. That plaque that builds up is a biofilm, and if we don't brush it off, it can lead to cavities and gum disease.
I don't even want to think about that. You also mentioned how biofilms can contaminate medical devices, causing infections that are hard to treat.
Yes, they can form on catheters, implants, all sorts of things. It's a big problem in healthcare. And because of that structure, biofilms are very resistant to antibiotics.
So it's not just about developing new antibiotics, but also finding ways to prevent biofilms or disrupt them somehow.
Absolutely.
I think it's safe to say I won't be looking at soil or even brushing my teeth the same way ever again. It's mind-blowing to think about this vast, invisible world of prokaryotes all around us, affecting us in so many ways.
I know.
We really need to change how we think about bacteria. They're not just germs to be wiped out.
Right. There's so much more than that. They're crucial for life. They can help us solve all sorts of problems, and they're always changing the world around us.
This deep dive has been incredible. I hope our listeners now have a better understanding of the amazing world of prokaryotes.
Me too.
They really are the unsung heroes and sometimes villains of life on Earth.
Well said.
I want to thank you for coming on the show and sharing your expertise with us. And to our listeners, thanks for joining us on this deep dive into the world of prokaryotes. Until next time, keep exploring, keep learning, and don't forget about the power of the microbial world.